Hi, everyone. My name is Nick, and today I'm going to be giving you a talk on my life as a virologist, which is the type of scientist that studies the viruses that make us sick. And what you'll find from this talk is that science is something that is a lifelong journey, starting from where you are right now, where I was not too long ago in grade school, all the way through college, and then, if you really love it, for the rest of your life. So, I would like to start my talk by telling you about the days where I was in the seat that you're in right now, because I started my science journey way back in high school. So, to tell you a little bit about myself, I am now 24 years old, and I'm in the second year of my PhD, a PhD being the type of university degree where people call you doctor, but you can't see any patients. It's a research scientist title. Um, I am a virologist by trade. As I said, I study viruses, but in my off time, I am a full-time nerd. I enjoy building custom PCs, playing role-playing tabletop sims. Uh, I like petting other people's dogs. Huge fan of that. Um, and you can see that I have been doing science for quite a while now. So here in 2014, this is me with one of my first science fair projects. Uh, and now this is a photo from me a couple months ago when I was 23 at a virology conference. So how did this happen? Where did this story come from? And my journey with science kind of began as a result of my old biology teacher. And yes, I am going to give him all the credit for this. I really do believe that a good teacher makes the difference for what you do for the rest of your life. So, any teachers who view this, you guys are the best. And this man, Jeremy Graney, got me into science by forcing me to do something I did not want to do. And that was my first science fair project. So this was freshman year of high school. This is a requirement for my intro bio class that we all had to do an independent research project. And what I chose to do for my very first research project, and I totally did not do this all the night before it was due, was to take orange seltzer water, which is clear and slightly orange flavored, add different amounts of orange food dye to it, and ask people, how do you think this tastes? Same seltzer, different color. And we humans, because we're not the brightest animals, consistently seem to find that the darker the color, the better it tastes, apparently. And even though I rushed this entire project for a due date, I found that I kind of enjoyed formulating my own questions, not just reading them out of a textbook, and then testing them out. And so, although this was a mandatory science fair, for the rest of my time in high school, I started deciding to do them on my own, just as like a club or a hobby. And I kind of started to up my game with my science fair projects. And the next idea I had to do with a topic that was happening right near where I lived in Rhode Island. And it was about this pond I have marked on a map. And this pond was going through something called an algae bloom. And an algae bloom is when algae start explosively just reproducing in the water, turns the water really green and gross, and it kills all of the fish life that exists. It's an environmental disaster that happens pretty often. And it's usually caused by pollution in the water that allows the algae to reproduce more than they should be able to. And I decided that I would get to the bottom of an algae bloom that was taking place in this lake near my home. And to get to that, I was testing for pollutants all around the lake to see if I could find a source of what could be causing this algae bloom. And those pollutants I tested for, they're just chemicals called phosphate and nitrate. They're usually found in fertilizer for people's lawns. But I also made a point of taking samples at different regions around the edge of this pond, because one side was bordering Rhode Island International Airport, TF Green. So I would sample directly from discharge places where the airport was dumping waste. And I'd also test for things like de-icing fluid, which they spray on airplanes and can leak into local ecosystems. And when I did this project, I got the interesting result that there were actually more pollutants on the airport side of the pond that could be implicated in algae bloom. And this made a lot of sense because the airport was undergoing construction, digging up a lot of soil, turning up a lot of fertilizers, which could run into the pond and really cause some issues. And so this implication of a specific entity and causing this environmental disaster took me pretty far at science fair. This is me when I was 16, my first science fair win, where I won the Rhode Island Fair and then was sent over to Phoenix, Arizona to present my research at the International Fair. And science can take you pretty far, obviously. It also can take your high school biology teacher pretty far. And he came along for the ride every step of the way. Um, and so this was great. This was like an incredible experience getting to do my first real research and feel real impacts of what's happening here. And it just led to even more ideas, as science always does. I detected a little bit of de-icing fluid in the pond on the airport side. And I was wondering how would algae respond to this chemical, which they spray on airplanes to get rid of ice when it's in their environment. And you might think that something like a chemical de-icing fluid is just going to be poisonous to anything that's in the water. Things in the water are sensitive to pollutants, right? This is supposed to be bad for them. Well, it turns out when you grow these algae, and these photo credits go to me, very proud of these when I was 16. When you grow algae in the presence of this chemical de-icing fluid, they actually grew multiple times more, which is a finding that was pretty novel at the time. No one really knew that this de-icing fluid could be used as a food source. And this is totally unexpected. I was expecting to figure out how toxic this de-icing fluid was to the algae. But in science, it's often the unexpected things that are the most helpful and illuminating things that can happen. And this led to yet more science fair victory. And this one led to some far-reaching travel to another international fair, the Stockholm Water Prize. And really, this just went so much further than I ever thought it would. As far as I was concerned, I was just a kid who was trying to find something to do in his downtime, came up with some questions, and then got some lucky breaks. But the spirit of science is that we're constantly questioning what's happening. And when you find things like this, it's what pushes us forward. And so I had logged all this time working with algae as a high schooler. And you might think, well, that's what I'm going to stick with forever. And the fact of the matter is, as soon as I went to college at Emory University, I immediately ditched algae. Another life lesson you can take here is that in science, you can always change your mind about what you're interested in. And the gear I shifted to from my algae research was influenza virus, the virus that comes around every season. You have to get vaccinated for it every year, or you should get vaccinated for it every year. And it causes pretty bad respiratory illness, coughs, sneezing. And I've stuck with influenza, even into my PhD, as my true blue research passion. So what's my work like with influenza? So as I showed here, this is kind of like a pretty picture of what an influenza virus looks like. This is a real picture of what an influenza virus looks like, not an animation. And flu viruses are really, really small. They're so small that we need special microscopes to even see them at all. And what we see when we take a picture of influenza is that the virus has this weird thing where it can take multiple different shapes. It can kind of be shaped like a circle, like this event that's pointed out here, also like the diagram. And it can be shaped like these giant long noodles. And I like to think of this as kind of like spaghetti and meatball flu. You have some that are small balls and some that are long noodles. And you might be wondering, well, why do we care about this? Why do I care if flu can be a noodle or a meatball? And the flu's shape actually has really, really big consequences for the disease within us when it infects humans. And the way this works is you can think of an influenza virus as a little ball that's covered in keys. And you can think of our lungs, or the cells that our lungs are made of, as locked doors. The virus wants to get in and infect. So it has these keys on its surface that unlock the door and let it in. When our body is fighting against a virus, it makes antibodies. And these antibodies are just little tiny things that stick to the keys on the surface of the virus and prevent it from doing anything. The key has this thing on top of it. It can't enter the host cell. It's blocked. Well, when flu takes a noodle shape, it's a much bigger virus and it has more keys on the surface. And if it has more keys on the surface, it needs more antibodies to block them all. And sometimes you miss a couple and this virus can still infect. And this is how the flu fights back against our defense in the form of antibodies. So understanding how shape works is a really important thing for our understanding how to treat these viruses and overcome these big noodle flu's ability to escape from our defenses. So how do we go about understanding shape? How do we crack the mystery of what decides what shape a flu virus is? Well, I'd like to make an analogy here. If you consider a house, before it exists, has instructions in the form of a blueprint. And if we make changes to this blueprint, then the house is going to look different. For example, maybe the chimney changes color, or maybe we put a bush outside. And this is from changing the instructions for building the house. Likewise, a virus has instructions to make a copy of itself, little tiny instructions how to make another virus. And that every virus contains a set of these instructions. And we think that within these instructions to make a virus, somewhere in these instructions, which are really complicated, there's something that's telling the virus whether to take the shape of a noodle or a meatball. And what we can do is, with really, really creative chemistry, we can make changes to the instructions to the virus and see, do we have more noodle flu? Do we have more meatball flu? How does this change affect the virus's shape? And this is how we understand how viral shape works. Except, unlike a house blueprint, a house is 10 meters long, the flu virus is about 100 nanometers long. So you could stack a few thousand of these, across the head of a pin. And it's just incredible how this chemistry is so controlled, we're able to do it on a meaningful basis. And then there's the flip side of things, that maybe the shape of flu isn't just encoded in the instructions for the virus. Maybe the flu changes with its environment, kind of like how we change when we go in the pool for a long time and our fingers get all wrinkled. The shape changes, but there's nothing about us that really changed on the basic level. So maybe with the flu, when it gets attacked by these antibodies, it's changing its shape in response. Different environment, different shape. And we have a feeling that it's a combination of these two factors that's really causing the phenomenon. And we're investigating both of these very rigorously to see if we can unravel this mystery. And of course, we want to understand how flu shape works. And we also want to be able to fight against the flu. We want to use our understanding of shape to create new treatments. And at Brandeis, we have an idea for getting rid of these noodle flus that require so many antibodies to get rid of the keys, and just making a big cage around them. Can we make like a force field around these viruses that completely surrounds them and stops them from infecting our cells? And I think we can engineer that material that forms an antivirus shell. And the way we do that is by making these little tiny molecular machines, so small it's hard to even imagine how small these are, that stick to our viruses, our virus being in yellow here, and assemble like magnets into a cage around the virus, a little jail cell. And what's great about this work is that we can make jail cells of all different sizes, from small to large, to stick to a virus of any type and any shape, to try and neutralize them and create a more comprehensive defense against these viruses as they infect us. And don't just take my word for it, because we can actually see these cages as they assemble in real time. So in these pretty movies I've shown here, we have a screen with a ton of different little building blocks for our cages, and we can watch as they all come together and then stick in these larger cage assemblies. And if we want to, we can make it go the other way. We start with our assembled cages, and we can have them come apart and clear from the system just like that. And I record these movies myself. I'm very proud of them. I think they're beautiful. And I just think it's so incredible that what we're looking at here is a world so tiny, it would not be possible to look at anything smaller than this. This is the ability of science. This is the world that we can look at through the lens of scientific methodology. Oop. And that kind of summarizes the things that I work on as a scientist. And I just want to wrap this up by saying that I told you a lot about how I did science fair as a kid and how that got me into it. But science isn't just for people who are making posters when they're 15. Science is for everybody who wants to get into it. I completely changed what I was doing when I went to university. I ditched my high school research and I shifted gears completely. And it's never too late. It's never too late to learn it. It's never too late to get into it. It's like playing an instrument or learning to paint. The more time you do it, the more time you spend around it, the more it just feels second nature. It might seem intimidating and unapproachable at first, but as soon as you jump in and you take that first step, everything just kind of falls into place after that. So hopefully I've convinced you that being a scientist is cool. I'll wrap up this talk now. For anybody who's interested in the work I do or wants to get more specific about how the science works, you can always email me at the address displayed here. And I just want to say that science is a great thing. It's fun to do. It's a great career. And if any of you have any curiosity about doing it, I highly encourage that you look into it more, talk to me, and I'll sell you on what a great idea it is to become a lab scientist. So thank you for listening.